Hi students, in the last lecture we have seen the structure of anther. When I was explaining the structure of anther, I told you there are 4 pollen sacs in a dithecous anther. Inside the pollen sacs, sporogenous tissue or microspore mother cells will be there. So these microspore mother cells, from that the pollen grains will be forming, and that process is known as microsporogenesis. Today we are going to study about microsporogenesis, how microspores or pollen grains are developing from a pollen mother cell, and then from that how the male gametes are developing, that we are going to study. So each microspore mother cell, it will be diploid. Okay, this diploid cell undergoes meiosis. Already we have studied about meiosis in 11th standard. Meiosis means it is actually from a diploid cell 4 haploid cells will be forming. Okay, it is known as reduction division. This all we have studied. Here from a diploid mother cell, haploid microspores are developing. Okay, how many are developing? 4. Okay, 4 haploid microspores or pollen grains, and as there are 4 microspores developing, we call this as a tetrad. Already this point I have told you in the last video. Now we have to see the structure of a microspore, and then how it is developing into a male gametophyte. Okay, structure of a microspore. Microspore, it is non-motile. Non-motile means not able to move. Haploid, already we know it is haploid. Okay, it is a gamete, male gamete, it is haploid. Then it is unicellular. Of course it is unicellular, and it is having a single nucleus. So a non-motile haploid unicellular body with a single nucleus, that will be the structure of a pollen grain or microspore. Now how it is actually protected? What is the outer wall? It is called, it is known as sporoderm. Externally it is surrounded by a two-layered wall known as sporoderm. The term you are supposed to remember, sporoderm. It is 2-layered, and in the 2 layers, the outer layer is known as exine and inner layer is known as intine. Okay, outer one is exine, inner one is intine. First we will see about exine. Exine, it is made up of a complex non-biodegradable substance. That substance is known as sporopollenin. Sporopollenin, it is actually very much strong, complex, not reacting, inert substance. Okay, it is non-biodegradable, it is very complex, and it is resistant to chemicals. This sporopollenin, you are supposed to know the peculiarities of sporopollenin. In your textbook there is a question given, why pollen grains can remain well preserved as fossils. It is given actually with this structure, the same page it is given. The question actually, it can be asked, why pollen grains can remain well preserved as fossil. The reason is over here. Okay, the peculiarity of sporopollenin, that is the reason. It is a complex non-biodegradable inert substance, and it is very much resistant to chemicals, and this nature preserve these pollen grains actually beneath the soil for number of years. You know, fossil study or paleontology or fossil studies. Okay, fossils means the remains, dead remains of organisms. Through that fossil study or paleontology, we are able to know about our means past ancient means organisms. Okay, that way we are able to know the means evolutionary history all. So this paleontologist means they will be collecting pollen grains from beneath the soil to study about the ancient plants, because that way it will be getting preserved. That the reason is that because of this sporopollenin, it actually help to so it is, as it is inert and complex and non biodegradable, it will be there beneath the soil for a means a long number of years. So that way we are able to study about. Now, so this question keep in your mind, the answer also keep in your mind, it can be asked. Now next, germ pore. So here two layers are there for the wall of the microspore. Exine, it is very thick and it is protective, I told you. And in some areas this exine will be thin. That areas are known as germ pores. Okay, in monocote there will be a single germ pore. It is actually known as uniporate. And in dicots there can be three means germ pores, then it is known as triporate. So germ pores are the regions through which the microspore or pollen grain is germinating out, developing out. Through that the pollen tube is coming out. Okay, this germ pore. Now about the inner layer, that is intine. Intine is actually a thin layer inside this exine. It is made up of cellulose and pectin. That is the means structure, generally the structure of a microspore. Now we have to see the development of male gametophyte. How male gametes and male gametophyte is developing from a single pollen grain. And one more thing, actually I forgot, this exine, actually its structure is also varies according to different species. Externally it can be smooth, or sometimes it can be actually spinous. So that way the structure varies according to means the different, in different species different types of means exine can be there. Some plants it will be smooth, the pollen grains will be smooth. Some plants there will be spines. Actually shoe flower roll, when we do the practicals, we can see there will be outer actually spinous pollen grain. When we see under the microscope, it is visible. Okay, now coming to the development of male gametophyte. Male gametophyte, how it is, there are two mitotic divisions take place. So already one meiosis got over, and by that meiosis actually 4 haploid microspores or pollen grains are forming. Now this haploid microspore, it undergoes 2 mitotic divisions. Mitosis also we have studied. Okay, equational division. So there are 2 divisions. First we will see the first mitotic division. Okay, in first mitotic division, this means microspore or pollen grain divides into 2 unequal cells, a large vegetative cell and a small generative cell. Okay, large vegetative cell and a small generative cell. Now this vegetative cell, it is rich in food and having irregular shaped nucleus. Okay, and generative cell, it will be actually small in size. It is having a large nucleus, but the cell will be comparatively smaller to the vegetative cell, and it will be floating in the cytoplasm of the vegetative cell. Okay, now this way 2-celled structure it is forming by the first mitotic division. Now second mitotic division, sometimes it take place within the microspore itself. Okay, or sometimes it will take place after pollination in the pollen. So second mitotic division in some plants, it will be taking place inside the microspore before pollination itself, or some plants it will be taking place after pollination. In the pollen tube we will see actually how it is happening with the help of the diagram. Okay, anyway we will go to the next page where diagram is one. See here the structure of the microspore it is given. Okay, outer exine and in between actually irregular intervals you can see the germ pore. Okay, this where it is getting the exine is thin, that area is known as germ pore. Okay, and inner thin layer it is intine, that is the cytoplasm is there. This is after first mitotic division, a small generative cell and a large vegetative cell. Okay, this way two-celled structure. So in some plants in this two-celled structure only pollination take place. In angiosperms most of the plants actually in this way two-celled structure they will be getting pollinated. You know pollination means, okay, from the anther the pollen grains are actually falling into the stigma of the means carpel. Okay, female reproductive world. So that is pollination. So after pollination, on the stigma, you know already the structure of flower. On the stigma there is a sugary substance. So these pollen grains will be absorbing that sugary substance and they will be, that way the cytoplasmic content all increases. Okay, then there will be a pressure created inside the intine and it will be with the cytoplasm, the intine will be growing out through the germ pore. So it will actually this way the pollen tube actually means coming out, the cytoplasm, the generative nucleus, tube nucleus all will be there. Through this germ pore it will comes out in the form of a tube. This tube is known as pollen tube. Inside that actually there will be a tube nucleus and a generative means nucleus or generative cell will be there. This generative cell actually it undergoes the second mitotic division inside the pollen tube. Okay, in some plants, or some plants it will be just means before pollination it can take place in the microspore also. So these two male gametes, the generative cell divides mitotically into two male gametes. Okay, there will be two male gametes in the pollen tube, you can see here, and a tube nucleus will be there which is the nucleus of the vegetative cell. It get actually degenerated later, it is not having further role. The tube nucleus will be means getting it will be actually afterwards it will be getting disappeared or degenerated. Okay, it is not having further role. But these two male gametes actually they both are having a role in fertilization, that we will study during fertilization. So structure of male gametophyte, if it is asked, don't forget there should be a two male gametes should be there, a tube nucleus should be there and a pollen tube should be there. Okay, so this way the structure and development of male gametophyte, if it is with the diagram you are representing, then all these stages you have to draw. First of the microspore, then the two-celled structure, and this generative cell it will getting floating in the cytoplasm of the vegetative cell, and then generative cell divides into two male gametes. Okay, and then the cytoplasm with the intine with this cytoplasm all it is getting flowing out through the germ pore by absorbing the sugary substance on the stigma. It is actually getting more means the quantity is increasing, and that way the volume is increasing. The pollen tube is growing out carrying two male gametes and one tube nucleus. Okay, both the male gametes they are involved in fertilization. That fertilization we will study later, and this tube nucleus get means degenerated later on. So this is the structure of male gametophyte. So for the structure and development gametophyte. These are the processes what take place. So here the male reproductive part of the flower we have completed. Next will be the female reproductive whorl of the flower, that is gynoecium. So about that in the next video explaining. Go through this video. If you have any doubt, please ask, and we will be going further in the next video. Okay students, bye. See you in the next video.